expression of both traits equally, and one sort of goes hand in hand with the other. So this is really, really well documented in flowers in particular because flower colors are really fun to play with. So people work with this. But you know, fruit fly research, there's kind of this stuff going on as well. But this is, why do you get this lilac-colored flower as opposed to either being, well, those are supposed to look like they're pink. Up there, they look purple to me. But so if you get sort of this dark allele versus the white allele, when you put them together, they show a pink condition. That's basically just showing that the heterozygotes themselves are expressing incomplete dominance in those alleles. And so, in all actuality, if you're using the term R, if you're using the allele code R and small r, when things are incompletely dominant, you sort of want to say, okay, it's not really against large R, capital R versus a lowercase r, right? It's not really because one's not dominant over the other. But there's no real way to sort of express that clearly because when they're together, they're completely different. So the homozygous dominant versus the homozygous recessive is very, very different look to them. It just happens that when they get together, they sort of struggle who gets to be expressed and who doesn't. And in some cases, incomplete dominance can be hidden just based on the environment as well. In certain types of soils, maybe the purple color doesn't come through quite so well, or the pink color doesn't come through quite so well, so you get something that's a little bit more like white when they cross. You never know until they're actually tested. But in general, it's incomplete dominance really just that neither allele really is dominant over the other, and pretty much your genotype and your phenotype are always going to show up in that 1 to 2 to 1 ratio. You're going to have 1 out of the 4 that's completely dominant for one color, 1 out of the 4 that's dominant for the other color, which we call the recessive trait, and then you have the two heterozygotes that are showing up out of the mix are showing both traits at the same time. And again, if you go back to this Punnett square, and you cross two heterozygotes, if you cross two purebred lines to make an F1 generation, 50% of them are heterozygotes, 25% are homozygous dominant, 25% are homozygous recessive, and so you can see this 1 to 1 phenotype matching the genotype itself. So you just go back to that original crossing and what you're going to get, and those numbers, those ratios should show. Okay, now we have to talk about codominance, which is another issue that everybody gets confused.